Welcome back. As we continue to work through our genetics or our DNA aspect of our semester here, last chapter, chapter 10, we really dealt a lot with our DNA to RNA to protein, that whole protein building process and how DNA works to serve as that control mechanism in our cell. Well, our next big topic then here in chapter 11 is looking at how are genes controlled. What determines when a gene should be activated or turned on and get turned into protein versus what may result in turning a gene off so that that DNA may exist in a cell, but the cell's not going to make any protein, and effectively then it's going to inhibit that particular gene. So as we mentioned last chapter, that gene expression is the process of going from our flow of information, DNA to RNA to protein, or when your genes get expressed, showing up in your phenotype. And lots of control aspects here in terms of when a protein should be made, how much of a protein should be made in a particular cell, and certainly that'll be our focus here today, looking at some of these control aspects. And we know that control of expression, or how much protein a particular cell makes, can be tied to a lot of different things. It may be tied to the environmental changes, as in the environment changes around a cell may respond by producing different proteins and have different gene expression. Different types of cell in your body certainly exhibit differential gene expression. A skin cell, for example, makes a completely different set of proteins than a blood cell or a nerve cell or a cell in your liver, for example. And when scientists study the gene expression and this control aspect, one area that was typically started is looking at prokaryotic cells, especially bacteria. Because bacteria have less DNA in their prokaryotic cells, they don't have that nucleus, remember, and they're very easy to grow in lab. They're very much a model organism, and certainly much of what we know about genetics is based on bacteria first, and then we take that knowledge and try to apply it to our more complex eukaryotic cells. And when people first started studying gene expression, they discovered what were called operons in these prokaryotic or bacteria cells. And an operon is a whole group of related genes, maybe as many as 3 to 5 different genes, all located near each other on a chromosome, and they all controlled in a coordinated or sort of singular fashion, meaning all those genes are either going to be expressed at the same time and turned on, or they may all be turned off. And the first of these that was discovered and really well understood is what's called the lac operon. And the lac stands for lactose, and that's a type of sugar. We know that lactose is the major sugar that's found in milk, for example. And so this lac operon has to do with the ability of the bacteria to degrade or eat this lactose sugar and turn it into usable energy. And so there are 3 different genes located in sequence next to each other on the bacteria chromosome, and these genes are what allow this cell to then metabolize our lactose sugar here. Well, it turns out that each gene is not regulated separately, but there is a single promoter sequence or sequence on the DNA, right in front of these 3 genes, and it's that promoter sequence then that's going to control whether or not those genes get activated and turned into RNA and protein, or whether they remain off. And so there seems to be a key chemical called a repressor that can bind to that sequence and block the expression of the gene. And so if the repressor is present, the genes are turned off. If the repressor is absent, then those genes are activated, and that cell then could metabolize its lactose sugar. So if we take a look at the lac operon in more detail here, we know that there's a gene that codes for this repressor protein. It's just called a regulatory gene, and certainly cells have many different regulatory genes that code for different repressors. And again, when there is no lactose around, that regulatory gene is going to produce that repressor protein. It's going to bind to the DNA and prevent our transcription. Basically, remember, transcription is our first step, our DNA to messenger RNA stage here. Okay. And so here's a picture just showing that. We can see here's our lac operon, our regulatory gene, then we've got our promoter sequence, the operator, which is that yellow sequence, and then our 3 genes that are going to allow the cell to use that lactose sugar. And so when that regulatory gene undergoes gene expression, it gets turned into RNA, into protein, and that repressor then is going to bind to the operator. And normally the operator sequence is where the RNA polymerase would bind and start converting that DNA into RNA. And so really the operator is the key starting point for transcription and gene expression. And so by that repressor protein just physically being in the way, it's going to shut down all of those genes, and that cell then will not produce any proteins associated with lactose metabolism, and it will not be able to utilize that sugar. But the chemical environment that the bacteria is in certainly can influence then the expression of this lac operon. And so, for example, if a bacterial cell is growing and lactose sugar is present, then what happens is that repressor protein that normally, as we saw, was binding to the operator, it was turning it off, that repressor protein actually will bind to lactose. And lactose will chemically react with the repressor protein if it's present, and that chemical reaction then changes the shape of the repressor, so it no longer will bind to that operator region. And so the repressor then is inactive, it no longer can stop the transcription of this lac operon, and so now these genes are active, that RNA polymerase will bind to the operator, and it will convert all those genes into protein and express them. And here we can see that visually on this little cartoon. Again, the repressor normally can bind to the operator, but if lactose is present, it's going to bind to that repressor and basically shut it off so it no longer works, which means now that lac operon is turned on. And so here's a good case where the environment certainly influences gene expression. The presence or absence of lactose directly determines whether that bacteria cell can metabolize or utilize that lactose for energy. So environmental influence certainly has a big impact on gene expression. Another area where we see gene expression playing a big role is in cell specialization. So if we think of multicellular organisms like ourselves, we know that we all started off as that single-celled zygote when egg and sperm fused together, and that cell was a stem cell, a basic cell type. It really wasn't specialized yet. And that as our cells develop over time within the embryo and eventually becoming adults, that our cells gradually through several small steps become more and more specialized, meaning we've got over 100 different types of cells. Each cell has its own structure and its own function. Well, the difference between those different cell types in our body are due to differential gene expression, which genes are being turned off and which are turned on. And so, for example, if we think of muscle cells here, we've got some smooth muscle cells with their striations. We've got some cells in the pancreas here. Or we look at something like a white blood cell as another example. Remember, all three of these cell types are all genetic clones. They're all exactly the same because they were all produced through mitosis. Okay? But what makes that muscle cell different than that pancreatic or that blood cell is gene expression. That each of those have a different set of genes turned on or active, so it's the proteins that are present due to this differential gene expression that make each cell type different. And a good example of this gene expression and determining different cell types is looking at fruit fly and fruit fly development. And fruit flies are used very similar to bacteria because they're easy to grow in lab and they're a very nice sort of study organism. So we know a lot about them. And what we see with gene expression in fruit flies is that differential gene expression is going to impact a bunch of different areas within development. The first thing that really is affected is the different ends of the fruit fly as the embryo develops, orientating the head versus the tail. And it turns out that there's messenger RNAs present in the egg produced by the maternal side, by mom, that are going to be expressed that are going to influence this development of which end is going to develop into the cells that become brain cells versus which end are going to develop those tail cells. We know that many organisms have segments to their body, and certainly fruit flies are no exception here, and it's protein products from a very specific set of what we might call segmentation genes that are producing the right proteins that cause this segmentation. And it turns out that there's a set of genes that sort of activates or turns on these different second set of genes then that are going to produce the segment. So again, it's all about gene expression. Which genes are being turned on determines then where the segments are going to form, and eventually which segments are going to develop into what body parts when we start thinking about something like, again, a fruit fly. And then if we think of adult features, it turns out there's a set of genes, and this set of genes is conserved really throughout most animals, ourselves included, and these genes are called homeotic genes. And homeotic genes are sort of the master control genes that determine large structures in developing animals. And it's these homeotic genes that are going to largely determine which body parts are going to develop on which segment of something like a fruit fly. And again, certainly we have some of these homeotic genes that determine our large structures like development of arms and legs and those sort of major structures within an organism. So here we can see just a figure showing some of the examples we talked about. Again, it's differential expression of messenger RNA into protein that's going to determine the head region of the egg, and the other end then is going to develop into the tail. It's the segments being formed by this differential gene expression that are going to result in the segmentation or the separation of the different body parts of that developing fruit fly. And again, as we think of adult fruit flies, then the location of cells in these different segments are going to determine what body parts are going to be made, what cells produce which proteins, and again, that's largely based on some of these homeotic genes. So these homeotic genes play very major structural roles in most animal groups, and they're going to control, again, where and how large body structures are going to develop when we think of legs and arms and antennas and eyes and ears and those types of things. And small mutations or small changes in those genes may have a big impact then in where or how many of these different body parts develop. And for example, oftentimes if we look at two organisms, we might think, well, those two organisms are really different. This organism has 6 legs and this other organism has 10. That just doesn't, they're just too different from each other. But we come to find out that a small mutation in a homeotic gene results in extra structures being built. And so actually they may be very closely related genetically, and it's a small change in these genes. And so, for example, your book uses this example again of the fruit fly, and one of the segments on the fruit fly are going to develop into the antenna. So on the forward part of the face there, we've got the large eyes on the side, the antenna pointing out. But a mutation in a homeotic gene results, instead of antenna growing there, legs growing there. And obviously that's a really weird, odd sort of thing, and certainly that's why the book used it as an attention grabber here, legs sticking out of the head here. But again, it seems like that's a really big genetic difference that would cause antenna versus legs, but in reality, it's one simple mutation that we have antenna producing proteins and we have leg producing proteins, and one switch of that homeotic gene results in the wrong proteins being built in the wrong place, and so whole legs develop instead of antenna. So again, sort of our master control developmental genes are these homeotic genes. So we mentioned that lack operon in prokaryotic cells, groups of genes regulated by a single promoter and operator system and repressor. But if we look at our eukaryotic cells, our cells are going to be a little bit more complicated. We have a lot more DNA, many more genes, a nucleus, and so on. And if we look at our genetic sort of makeup, what we see right away, first of all, is we don't have operons. Eukaryotic cells, we don't have groups of genes clustered together and all controlled by a single promoter repressor region. Instead, in eukaryotic cells, each one of our genes has its own promoter and its own termination sequence, so that a single gene is going to be expressed or not expressed, as opposed to whole groups of genes like we saw with that operon. And there are lots of different factors in our cells that are going to regulate this gene expression, regulate which gene is going to be active and which gene is turned off. And some of those things include the physical packaging of the DNA. So how your DNA is actually physically, chemically packaged in the cell may impact whether a gene is going to be active or inactive. We can switch on or off different genes by what are called activators or promoters that are, again, going to be chemicals that may bind to your gene that are going to allow it to be turned into RNA and eventually protein. Okay. Or there are several different types of other regulatory chemicals that may either act to increase the expression or activate a gene, or other of these regulatory chemicals may act to decrease or suppress a gene and turn a gene off. And so we're going to take a look at some of these different regulatory mechanisms in eukaryotic cells and just a few examples of them. And as we look at this regulation of gene expression, one thing we're going to see is a single gene may be regulated by one of these different types of mechanisms, or maybe a combination of 2 or more of these different mechanisms, just depending on the particular gene. And so if we take a look at some major categories, chromosomal changes, or again the physical structure of how the DNA is packaged, may impact expression, and so DNA may be packed or unpacked to turn off or on a particular gene. Transcription, remember our DNA to messenger RNA step of gene expression may be regulated by different control proteins, either to activate or to suppress that gene. We may have the RNA being processed differently. Remember, our RNA gets processed, those introns are removed, and exons, we said, are spliced back together to make our mature messenger RNA. We also have a cap and a tail added, so whether that's added or how much RNA is added to the cap or tail may influence expression. Sometimes RNA may be broken down, and that's going to suppress a gene then if that RNA is chopped up into little pieces, for example, before it can be turned into protein. And we're going to see different chemicals may interfere with that messenger RNA and how it does get turned into protein. And then finally, we may have translational regulation. And translation, remember, was our RNA to protein step. And so there are other chemicals that may regulate or interfere with that step. And so basically, we can have regulation occur anywhere along the way, whether it's the DNA storage stage, it's DNA to RNA, it's our RNA processing, or it's our RNA to protein stage. Any of our different aspects of our flow of information may be regulated. So we're just going to go through and take a look at each of these and a specific example of each type. So a simple way to look at gene expression has to do with our DNA packaging. And we know that our DNA is in the form of that double-stranded helix. We've got two chains of our nucleotides, and they form that spiral sort of staircase shape. But our DNA is packaged in our cells, and remember, our normal form of packaging of DNA is in the form of chromatin. And in chromatin, we've got our double-stranded DNA, and it's kind of wrapped around these DNA packaging proteins, kind of coiled or wrapped around. And a nucleosome is formed when the DNA is wrapped around these specialized packaging proteins called histones. And so histone proteins make little sort of ball structures or packaging proteins, and the DNA is wrapped around those little balls. Okay? And we can think of it like beads on a string, because when we look at it under the electron microscope, each histone looks like a little bead, and then the string or the DNA is wrapped around that bead. Okay? On top of that, then, these nucleosomes will start to wrap around and be packaged together, and that forms a big helical fiber. These helical fibers then are wrapped around each other on top of that, which forms what's sometimes referred to as supercoiled DNA. And so there's a lot of levels of packaging here, from very loosely packed nucleosomes to tighter and tighter bundles of DNA, eventually forming these supercoils. And we know that when DNA is packaged up in this supercoil form, it's basically inactive, that the proteins to go from DNA to RNA can't bind to the gene and start transcription when it's packaged very tightly. And so one way that a cell can regulate its gene expression is by packaging up the DNA that will turn off the particular gene, or unpacking it into a more loosely packed form like the nucleosome form. Then that gene would be active. Here we can see that level. Starting with the left, we've got our double-stranded DNA. The little balls represent the histones. So a nucleosome is the DNA wrapped around several of those histone proteins. Those nucleosomes then get coiled together, and those coils then get spiraled together into our larger, tightly packed chromosomes. And again, DNA would be turned off in that form. And here's that idea of beads on a string, because under the electron microscope, sort of the clear or the white area represents DNA, and when it's wrapped around our nucleosomes, it makes little bumps or beads, and then the DNA in between the beads sort of appear like a little string holding things together. A special case of this idea of gene expression being influenced by packaging has to do with X chromosomes, and especially in females of mammal species, certainly including humans. And one of the issues we have, if we think about our chromosomes, excuse me, men have only one X chromosome and women have two X chromosomes. And so there's a problem here, because women would make twice as much of these X chromosome proteins as men do, because they've got two copies of everything. And so this is called a dosage effect. And so one of the issues we have to see is this dosage effect has to be removed. Our cells have to be able to compensate for this, either by males producing twice as much X chromosome protein, or by females producing half as much, to sort of level the playing field. And in mammals, the latter is the choice here, that females produce half as much X chromosome product, and the way that's done is one of the two X chromosomes in women is going to be inactive. And so while each woman has two X chromosomes, one of those two is going to be inactive, which means it's going to be packed up very tightly, just like we saw in that last slide, and basically all the genes on that X chromosome are going to be inactive or turned off because of that tight bundling. Now, if we were to look at a woman's cells throughout her body, what we would see is that it's kind of random inactivation, that some of a woman's cells are going to have the X inherited from dad's side of the family inactive or turned off. The other half of her family are randomly going to have mom's inherited X turned off. And so it seems to be kind of a random thing. And so women, actually, your cells are made up of a mosaic of cells. Half of your cells with dad's X turned off, half of your cells with mom's X turned off. Now, this X inactivation happens very early during embryonic development, and then, for example, if a cell has dad's X turned off, every time that cell divides by mitosis, all the cells that are produced are also going to have dad's X turned off. And so this happens very early on, and then this inactivation is passed on from cell to cell. Now, an X chromosome that's inactive is packed so tightly, it's bundled up so short and fat, it forms what's called a bar body. because it's visible under the light microscope as this little sort of chunk of DNA, and again, it's going to be inactive. So this would be our special case of packaging having an impact on gene expression. And a good place to observe visually this idea of X inactivation has to do with calico cats. And in calico cats, the two different colors in the fur, the orange and the black, are due to two different alleles of a particular gene that's on the X chromosome. And so in something like a calico cat, for example, a calico is always going to be heterozygous, having a copy of the orange and the black allele. And when those X chromosomes are inactivated, if the if the black color containing X is inactive, that means the orange fur color is active, and that's going to result in cells that produce orange color in the fur. Likewise, if the orange is inactive and turned off, that means the black containing X is going to be active, and so those cells would produce black fur. And so the mixture of the blacks and the oranges is going to be specifically due to the mosaic of different cells, depending on which X is active and which X is inactive. A second mechanism for regulating gene expression in our eukaryotic cells has to do with chemicals or proteins in particular that are going to influence transcription. And there are different types of these regulatory proteins that may influence our gene expression. One set of these proteins may actually activate or cause a gene to be expressed more often, causing the protein to be built at high levels. And those chemicals are called transcription factors. And these transcription factors will promote the RNA polymerase enzyme to bind to the DNA, again, to enhance transcription. But there are also chemicals called silencers that act as repressors or inhibitors that will actually bind to the gene and cause that gene then to be turned off, or that expression then to be limited in a particular cell. And so different types of regulatory proteins are going to act to either increase or decrease our gene expression. Okay? And in sort of upstream or ahead of genes, we have different control sequences. We have promoter regions, which is where the RNA polymerase is going to bind and eventually cause that gene to be expressed. And there are also several different types of enhancer regions. And these enhancer regions are similar or related to the genes of interest, and they can be controlled by similar chemicals. So certain similar groups of genes may be expressed in a similar pattern based on whether or not these enhancers are being produced or not. So if we look at an example of this idea of these transcription factors acting as activators or acting to promote or increase gene expression to turn a gene on, these activator proteins will actually physically bind to regions of the DNA, causing the DNA to bend in shape, which then allows that that RNA polymerase to bind to the gene's promoter region and cause the gene to be transcribed or to be expressed. And so it's a combination of these proteins being present that's going to increase the likelihood then of that enzyme binding to the promoter region and expressing the gene. If those transcription factors are absent, then the RNA polymerase cannot bind to that promoter region properly, so that gene would be effectively turned off. Another area where we're going to see this regulation occur has to do with lots of small RNAs and the expression of genes based on RNA processing and translational issues, the RNA to protein step. And if we just take a step back for a moment, if we look at the history of biology and our study of cells and the chemical nature and the kind of molecular study of genetics, we see that there have been several waves of how we've learned about different chemicals, and these waves are based mostly on technology issues. For example, the first wave that we really started to understand cells was to be able to understand proteins. And so we had sort of the protein phase of chemical biology or biochemistry starting in the late 60s and going throughout the 70s and mostly part of the 1980s, as people got the technology to be able to isolate proteins, to study proteins, to get the technology to sequence a protein so we knew which amino acids kind of made that protein and determined its shape. So we went through that sort of protein learning stage. The next stage then is the DNA stage. By the 1980s and throughout the 1990s and into the 2000s, then we got the technology to isolate DNA, to copy DNA, to sequence DNA and do genetic tests and so on. And so we've learned a tremendous amount about how DNA works in terms of the cell and building proteins. Well, we're just entering the next stage, and our next stage or wave is the RNA stage. Now, RNAs are very unstable, and they're hard to study outside of cells, but we're just now getting the technology to isolate RNAs, to copy RNAs, and to study RNAs. And so we went through the protein learning stage in the 70s, we went through the DNA learning stage in the 90s, and now in the 2010s, we're going through sort of this RNA stage. That when we're learning that RNAs, there are a lot more than we thought. They play a lot of different roles, especially in this gene expression issue that we're looking at. And there's lots of different types of these RNAs. A couple types I just want to mention that we know a little bit more about. These RNAis, which stands for RNA interference. And these are small RNAs that are going to interfere with the translation of RNA, messenger RNA, into protein. And typically what's going to happen with these interference RNAs is they're going to bind to their sort of matching messenger RNA. Remember, we have our complementary base pairing, and so these RNA interferences will have a sequence similar to messenger RNAs and will actually bind to the messenger RNA and physically get in the way, and that's not going to allow then for that messenger RNA to become protein. And so eventually then that messenger RNA would be degraded and recycled, never leading to a protein. So these RNA interferences can effectively suppress or turn off gene expression for particular genes. Another type of RNA we're just learning about are called microRNAs. MicroRNAs are extremely small, 20 AUGs and Cs long or less, and they're single-stranded. They tend to bind to messenger RNA and protein complexes, and they actually will chop up or dice up the messenger RNA and cut it up into lots of tiny little pieces. And by doing that, again, obviously that's going to prevent that messenger RNA from becoming protein. Now, our protein and DNA knowledge, because we've been studying it much longer, we know a lot more about it. Our RNAs, as I just mentioned, we're just starting to get into our RNA stage, so we're just learning about these, but there are lots more of these different RNAs, and many of these are going to play similar sorts of roles in terms of regulating our gene expression. And so these microRNAs, which are labeled in our picture here as miRNAs, they will bind to messenger RNAs, and there's different outcomes just depending on which protein complex binds to the combination there. Certain proteins plus microRNA will result in chopping up or degrading the messenger RNA. Others will result in that microRNA from sticking to that messenger RNA, which is physically going to then block the ribosome from translating that RNA into protein. Either way, the end result is the same. These microRNAs are going to suppress or turn off genes. A final mechanism for regulating genes then is to interfere with our translation, our last step here. And so if we look at gene expression, what we can see is, again, sometimes we're going to see that messenger RNA broken down, as we mentioned, with those small RNAs from our last slide. Sometimes there are chemicals that may block the initiation of translation. They're going to prevent the ribosome from binding to the messenger RNA, so that would turn a gene off. Other chemicals are involved in processing the string of amino acids and producing the final protein. And so some of these may result in protein activation, so they're going to increase gene expression. Others may result in breaking down proteins. And a nice example of that has to do with insulin. Insulin as a protein is made up of two separate polypeptide chains or chains of amino acids, and they start out as a single chain. When that chain is built, that long chain of amino acids is inactive, and if certain sulfur bonds are formed, and that S to S result is an example of a sulfur bond. If those sulfur bonds form, that insulin is inactive, but then this protein is going to be processed and chemicals will come through and actually cleave or cut those two strands apart, resulting in the active form of insulin. And so one way the gene expression of insulin could be regulated or is regulated is that if these cleavage proteins are present, then the insulin will be produced, so that gene will be active. If those cleavage proteins are inactive, that insulin will stay as kind of the round folded up form, the inactive form, and not produce the active protein. So this is the final stage then where expression can be either turned on or turned off. And your book uses this analogy, kind of putting all these different types together of kind of valves in the water pipe, that by turning on different valves or turning off valves, that's going to influence gene expression. And so for example, our first valve up there would be the packaging form of the DNA. By packaging up tightly, we're going to turn it off. By unpacking our DNA, that DNA can be turned on. Then we're going to have those transcription issues. whether transcription is activated by those transcriptional factors or it's turned off by repressors. It's going to sort of open and shut another valve. Then we may have issues with our RNA processing. Is that RNA going to be chopped up by those little microRNAs and RNA interferences? That would be shutting the valve off as well. Okay? But if the valves all stay open and the messenger RNA is produced and it's processed correctly and it makes it out to the ribosome, then it may undergo translation. And then finally, our final valve, whether or not we process the protein correctly, like with those cleavage example with insulin, or whether those proteins are turned off. And so by turning on and off these different valves, cells have multiple places then along this process of gene expression where it can be regulated, where a cell can activate that gene or inactivate that gene. So for example, let's take some of this information and just kind of apply it. And so I've got three just examples of what might happen in a cell, and I want you to think about each of these and how they would impact gene expression, how much protein is being built. So for one example, let's say messenger RNA, which normally lasts maybe at the most 24 or 36 hours in a cell, is more stable, and it was to last twice as long in the cell cytoplasm without degrading. How would that impact our gene expression? If the region of a chromosome that contains a particular gene becomes tightly wound or tightly compacted, how is that going to impact gene expression? Or if one of these cleavage enzymes was missing that's required to activate a protein, how is that going to impact gene expression? So if we think of these three scenarios, obviously the first one here, if mRNA becomes more stable, what would happen then is that mRNA would get turned into protein. It would undergo translation more often. And so that would be a case, that first example, where we would increase gene expression. We'd have lots more of that protein. The bottom two examples are going to act to decrease or turn off that expression. Because again, if the DNA is packed too tightly, that DNA then cannot undergo transcription, so it will be turned off. Or our bottom example, if our cleavage enzyme was missing, then even though the cell makes the chain of amino acids, it's never going to finish the protein and make the correct protein, and so effectively then that gene would be turned off. And today we certainly have the ability and the technology to measure gene expression, to look at different cells in our body and compare which genes are being expressed and turned on and which are being turned off. And that's done through what's called a microarray or a DNA microarray. And basically, in a DNA microarray, we take these very small kind of plastic trays and we fuse DNA from different parts of your genome all across this microarray into a big grid containing all of our different genes. And then to test for gene expression, what we would do is take a specific cell type. Let's say we're going to take a lung cell. And from that lung cell, we're going to isolate the messenger RNA. And of course, that would represent then all of the genes that are active undergoing transcription. From that messenger RNA then, in a test tube, we're going to treat it with chemicals that are going to cause that messenger RNA to be converted into DNA. And that's called reverse transcription, because remember, normally in our cell, our flow of information is DNA to RNA to protein. But in this artificial setting, we're going to take RNA and treat it with enzymes to make DNA. And so we're kind of going reverse in our normal flow of information. Okay? And when we make this DNA, it's called cDNA because it's DNA copied from messenger RNA. So that C stands for copied DNA. And if we do that with chemicals that are fluorescent, then when we make this new cDNA, it's going to be fluorescent, which means visually we can see it. Okay? Then we're going to take that cDNA and we're going to mix it with that microarray grid. Okay? Now, if a gene is being expressed, then its cDNA copy should be present, and it will bind to its gene or DNA that's fused to the microarray. Because of our complementary base pairing, that cDNA should bind to the microarray. If the gene is not being expressed, there's no cDNA in that cell for that particular gene. And so when we look at a microarray, what happens is anytime that cDNA binds to a particular spot in the microarray plate, that tells us that particular gene is being expressed. And we can tell that because, again, the cDNA is fluorescent. So we can either look at it visually or put it under a scanner that's going to measure that fluorescence and tell us which genes are binding to the microarray and which genes. And here we can visually just see this process of this microarray. And so again, we're going to have our microarray with each gene kind of fused to different wells or different parts of that tray. We're going to take our cell type, isolate the messenger RNA, and then using reverse transcriptase and fluorescent chemicals, we're going to make a copy of that cDNA. and then we're going to allow that cDNA by treating it with the right chemicals and the right temperature regime to bind to the microarray. Wherever it binds, we know that's an active gene. Wherever it doesn't bind, we know that gene then is inactive. So to summarize some of our gene expression issues here, we know that prokaryotes regulate their genes by the use of operons, and we looked at that lac operon example, that we've got a repressor and promoter system that regulates several genes at a time, and certainly in prokaryotic cells there are other operons as well. This is just the best understood. That cell specialization is due to differential gene expression in multicellular organisms, and we looked at that example, those homeotic genes playing a major role in development of different cell types and different structures within an organism. Then we talked about lots of different mechanisms of control, whether it's the packaging, controlling transcription, processing, or translation. Lots of ways your cells can regulate which genes are turned on and which genes are turned off. And then finishing up, we took a look at those microarrays as a way that today, a technology that we have where we can actually measure and look at which genes are active and which genes are inactive.